Hello, friend. Tonight we're going to slow down and gently explore something extraordinary, your own biology. Together, we'll unravel the secrets of genetics, not to study, not to memorize, but just to listen, to wonder, and to rest. Let the science carry you softly into sleep. Chapter 1. What is genetics? A quiet introduction to life's code. When we look at the natural world, from the delicate structure of a single leaf to the intricacies of the human brain, we see complexity that seems to border on the miraculous. Yet beneath all of it lies a quiet, invisible system of instructions, a biological language that defines, guides, and maintains all living things. This language is not spoken or written but encoded in molecules. It is the foundation of a field called genetics, the study of how organisms inherit traits, how biological instructions are passed from one generation to the next, and how the living world is built and maintained at the most fundamental level. At the heart of genetics is a molecule called deoxyribonucleic acid, or DNA. This molecule is composed of 4 chemical bases: adenine, thymine, cytosine, and guanine, arranged in sequences that resemble a long string of code. These bases pair up in specific ways: adenine with thymine, cytosine with guanine. The arrangement of these base pairs along the DNA strand forms the genetic instructions needed to build proteins and regulate biological processes. This sequence is not just a passive string of chemicals. It carries meaning, information, in the same way the order of letters forms words and sentences. But DNA doesn't exist in isolation. It's bundled into structures called chromosomes, which are located in the nucleus of almost every cell in your body. Each cell in the human body, except red blood cells and gametes, contains 23 pairs of chromosomes, for a total of 46. These chromosomes include around 20,000 to 25,000 genes, though the exact number is still the subject of ongoing research. A gene is a specific segment of DNA that contains the instructions for making a particular protein or set of proteins. Proteins, in turn, carry out nearly every function within a cell, from structural support and chemical catalysis to signal transmission and immune response. The human genome, which is the complete set of genetic material for a human being, consists of roughly 3 billion base pairs. Yet only a small portion of that DNA directly codes for proteins. The rest includes regulatory regions, non-coding sequences, and vast stretches of what was once considered junk DNA, now understood to have more nuanced roles in gene expression and genome architecture. What makes genetics particularly fascinating is its precision combined with its adaptability. The DNA in your body is remarkably consistent. Every cell in your body carries the same genome, with a few exceptions. Yet the way that genetic information is used can differ radically from one cell type to another. A neuron expresses different genes than a muscle cell, even though they both originate from the same zygote and carry the same DNA. This differentiation is controlled by gene regulation mechanisms that selectively turn genes on or off, depending on the cell's needs, stage of development, or environmental context. This phenomenon, known as differential gene expression, is what allows a single fertilized egg to give rise to the vast array of tissues and organs in a complex organism. Genetics is also the science of inheritance. When organisms reproduce, they pass genetic material to their offspring. In humans and most animals, this involves combining half the genetic material from one parent with half from the other. This mixing process creates genetic variation, which is essential for evolution and natural selection. Through mechanisms such as independent assortment, recombination, and mutation, every individual receives a unique genetic blueprint. Even identical twins, who begin life with the same DNA, can exhibit differences over time due to epigenetic changes and environmental influences. This inheritance of traits was first systematically studied by Gregor Mendel in the 19th century. Working with pea plants, Mendel discovered that certain traits followed predictable patterns across generations. He identified what we now call dominant and recessive alleles, versions of a gene that can influence how a trait is expressed. His work laid the groundwork for the field of classical genetics, although the molecular mechanisms behind inheritance remained unknown until DNA's structure was discovered nearly a century later. That breakthrough came in 1953 when James Watson and Francis Crick, building on the X-ray crystallography data produced by Rosalind Franklin and Maurice Wilkins, proposed the double helix model of DNA. This elegant spiral staircase-like structure explained not only how DNA stores information, but also how it can be copied accurately during cell division. When a cell divides, its DNA must be duplicated, a process known as replication. The two strands of the helix separate, and each serves as a template for the creation of a new complementary strand. The result is two identical DNA molecules, each containing one original strand and one newly synthesized strand. This fidelity is essential for life to continue through generations, though occasional errors, mutations, can introduce variability for better or worse. Understanding genetics also means understanding the context in which genes operate. DNA doesn't act alone. Within the cell, a host of molecular machines are involved in reading and interpreting the genetic code. Transcription is the process by which a gene's DNA sequence is copied into messenger RNA, a kind of temporary script. This RNA then exits the nucleus and enters the cytoplasm, where it is translated into a protein by ribosomes. Each triplet of RNA bases, called a codon, corresponds to one amino acid, the building block of proteins. The sequence of codons thus determines the structure of the resulting protein, and structure determines function. But the story doesn't end there. Gene regulation adds further layers of complexity. Promoters, enhancers, silencers, and other regulatory elements in the DNA help determine when, where, and how much of a gene is expressed. In some cases, non-coding RNAs can interfere with or fine-tune gene expression. External signals, from hormones to nutrients to stress, can also affect gene activity, highlighting the dynamic relationship between genes and their environment. This interaction between nature and nurture is one of the most profound insights genetics offers. You are born with a specific genome, but the way that genome is used can change based on your experiences and surroundings. In recent decades, our understanding of genetics has expanded dramatically thanks to advances in sequencing technology. The Human Genome Project, completed in 2003, mapped the entire human genome for the first time. Since then, the cost and speed of sequencing have improved exponentially, opening up new frontiers in personalized medicine, ancestry analysis, and evolutionary biology. We can now identify genetic markers for diseases, trace lineage across thousands of years, and even compare human DNA with that of other species to uncover our shared history. Yet for all its power, genetics is not destiny. While your DNA influences your physical traits, risk for certain conditions, and some aspects of behavior, it does not determine your fate. Environmental factors, lifestyle choices, and random events play equally important roles. Even traits that are highly heritable, like height or intelligence, are shaped by complex interactions between multiple genes and external variables. This understanding has helped shift the narrative around genetics from one of determinism to one of possibility. It is not a rigid script, but a flexible guide. At its core, genetics is the study of how life stores and transmits information. It reveals that every organism on Earth, from bacteria to blue whales, operates using the same molecular alphabet. The unity of this system, the fact that the same DNA language applies to all life, is one of biology's most humbling and awe-inspiring truths. It suggests a shared ancestry, a single thread woven through billions of years of evolution. Your genes are not just yours, they are echoes of ancestors, refined over eons, pass quietly through the bloodstream of time. And yet, for all its scientific clarity, genetics also invites wonder. That a molecule could store not only your eye color, but the code for building a human heart. That a silent string of chemicals could one day let you read these words. That within you, written in spirals and strands, is not just who you are, but where you came from, and maybe where life itself is headed. Chapter 2. The Double Helix, How DNA Stores Life's Instructions. In the microscopic depths of your cells, nestled within the nucleus, lies a molecule that quietly governs the operation of life. It does not make noise or emit light, and yet it holds the master instructions that shape who you are. This molecule is DNA, and its structure is as elegant as it is efficient. The double helix, now an icon of biological science, is not just a beautiful shape. It is the key to how genetic information is stored, copied, and interpreted. Understanding the physical nature of DNA helps explain how it functions as the carrier of life's instructions, and how it can replicate with extraordinary precision while still allowing for change. The structure of DNA was not immediately obvious to early researchers. For much of the 20th century, scientists were still debating whether proteins or nucleic acids were the material of inheritance. Proteins seemed more plausible due to their structural complexity, but that changed when experiments by Avery, McLeod, and McCarty demonstrated that DNA was the substance responsible for transmitting genetic traits. Building on these discoveries, James Watson and Francis Crick, with critical insights from Rosalind Franklin and Maurice Wilkins, published the proposed structure of DNA in 1953, a double helix composed of two complementary strands twisted around one another like a spiral staircase. Each strand of the helix is made of repeating units called nucleotides. Every nucleotide consists of three components: a phosphate group, a 5-carbon sugar called deoxyribose, and one of 4 nitrogenous bases. The bases adenine, thymine, cytosine, and guanine are the information-carrying components of the molecule. It is their specific sequences, like letters forming words, that encode instructions. The sugar and phosphate groups form the backbone of the DNA strand, creating a stable structure that protects and organizes the bases. The bases extend inward, pairing with complementary bases on the opposite strand. Adenine always pairs with thymine and cytosine with guanine. These pairs are held together by hydrogen bonds, which are strong enough to keep the strands connected, but weak enough to allow them to separate when necessary. The two strands of DNA run in opposite directions, a feature known as antiparallel orientation. One strand runs from the 5 to the 3' end, while the other runs from 3 to 5. This directionality is essential for many biological processes, including replication and transcription, which depend on enzymes that move in a specific direction along the DNA. The helical twist itself, with about 10 base pairs per turn, helps compact the DNA into a manageable space while still making it accessible to the molecular machinery of the cell. The double helix is not just a passive storage system. Its specific geometry plays a functional role in the cell's ability to access and replicate genetic information. When a cell prepares to divide, the two strands of DNA are separated by enzymes like helicase, and each serves as a template for the creation of a new complementary strand. This semi-conservative replication ensures that each daughter cell receives an exact copy of the DNA, preserving the genetic identity of the organism across generations. This process of replication relies on base pairing rules to maintain fidelity. DNA polymerases, the enzymes responsible for synthesizing the new strand, add nucleotides one by one, matching them to the bases on the original strand. They also have proofreading abilities, correcting mismatches before they become permanent. Despite the high accuracy of this system, occasional errors do occur. Some are corrected by additional repair enzymes, while others persist, becoming mutations. These mutations, though sometimes harmful, are also the raw material of evolution, introducing variation into the genetic code. The stability and reliability of the double helix are remarkable, but equally important is its accessibility. For the genetic code to be used, the information must be read, and this begins with the temporary separation of the two strands in a region of a gene. There, RNA polymerase binds and synthesizes a complementary RNA copy of the DNA sequence, a process known as transcription. The resulting messenger RNA carries the code out of the nucleus and into the cytoplasm, where it directs the assembly of proteins. The physical structure of DNA also includes regulatory regions, such as promoters and enhancers, that do not code for proteins themselves, but influence the timing and level of gene expression. These elements can be bound by proteins that either encourage or suppress transcription. Because DNA is a long molecule, about 2 meters of it packed into every human cell, its organization is vital. This is achieved through the wrapping of DNA around histone proteins, forming nucleosomes. These bead-like structures further coil and fold to create chromatin, which can be tightly or loosely packed depending on the activity of the genes in that region. This packaging is dynamic. During cell division, the chromatin condenses into visible chromosomes. But during interphase, the active period of a cell's life cycle, the DNA is more relaxed, allowing genes to be accessed for transcription. This structural fluidity is another layer of control, enabling the cell to respond to developmental cues and environmental signals. Another intriguing aspect of the double helix is its ability to transmit not just information, but memory. Epigenetic modifications, such as the addition of methyl groups to DNA or chemical changes to histone proteins, do not alter the base sequence but affect gene expression. These modifications can persist through cell divisions and, in some cases, even be passed to offspring. They represent a molecular form of memory, an echo of the cell's past experiences, encoded not in the sequence itself, but in its chemical context. The discovery of the double helix structure also had practical consequences that reach into modern medicine, biotechnology, and forensics. Knowing the physical nature of DNA allowed scientists to develop techniques to isolate, amplify, and sequence it. Polymerase chain reaction, or PCR, allows tiny amounts of DNA to be copied exponentially. Gel electrophoresis enables the separation of DNA fragments by size. Sequencing technologies can now read entire genomes in days rather than years. These tools are built on our understanding of how DNA is structured and how its strands interact. The double helix also offers an explanation for how inherited diseases arise. When mutations disrupt the sequence of a gene, the resulting protein may be malformed or absent, leading to a cascade of effects within the body. Cystic fibrosis, sickle cell anemia, Huntington's disease, each is tied to specific changes in DNA. Our ability to locate these mutations and understand their consequences is made possible by knowing exactly how DNA is constructed and how it behaves. There is an almost poetic efficiency to the double helix. It is compact but comprehensive, flexible but stable, specific yet universal. It encodes billions of years of evolutionary refinement in a form that can be passed effortlessly from one generation to the next. And perhaps most remarkably, it is built from just a handful of atoms: carbon, nitrogen, oxygen, hydrogen, and phosphorus, arranged in just the right shape to carry out the business of life. Despite its complexity, the double helix is consistent across all known forms of life. From simple bacteria to complex multicellular organisms, the structure of DNA is nearly identical. This universality suggests a common ancestry and hints at a deep connection among all living things. In a way, the spiral of the double helix is not just a molecular shape. It is a symbol of continuity, of memory, of a code shared by all beings that breathe and grow and evolve. To think that such a form, twisting silently in every cell, carries the instructions for making a heart, a thought, a dream, or a memory, is to be reminded that life is both mechanical and magical. The double helix is not alive, and yet it gives rise to life. It cannot think, and yet it stores the potential for thought. It is perhaps the closest thing we have to a biological infinity, repeating, mutating, replicating, enduring. You carry it in every cell, and through it, you carry the story of everything that came before you. Chapter 3: Genes and Proteins, from Code to Function. The language of life is not merely stored in DNA, it is translated, interpreted, and brought into action through a process that transforms inert sequences of molecules into the very substances that make up the body and regulate its every function. Genes, small segments of DNA carrying precise instructions, act as templates for the production of proteins, which in turn form the structural framework and operational machinery of all living organisms. Understanding how genes give rise to proteins bridges the gap between static information and biological activity. This transformation, from abstract sequence to functional substance, is the heartbeat of genetics. A gene is not just a passive stretch of DNA, it is a working blueprint. While the DNA molecule is composed of billions of nucleotides, only a small percentage of it corresponds to protein-coding genes. These regions are interspersed among vast stretches of non-coding DNA, some of which are involved in regulatory roles, structural support, or sequences whose functions are still not fully understood. When scientists refer to a gene, they typically mean a region of DNA that includes all the elements necessary to produce a specific protein. This includes the coding sequence itself, as well as upstream and downstream regulatory elements that help determine when, where, and how much of that protein is made. The journey from gene to protein begins with transcription. This is the process by which the genetic information encoded in DNA is copied into messenger RNA, or mRNA. This RNA molecule serves as a temporary copy of the gene that can leave the nucleus and travel to the ribosome, the cell's protein factory. Transcription is initiated when an enzyme called RNA polymerase binds to a specific region near the start of the gene, known as the promoter. This enzyme then moves along the DNA strand, reading its base sequence and synthesizing a complementary RNA strand. The RNA strand is constructed with ribonucleotides, which are similar to the DNA nucleotides but contain the sugar ribose instead of deoxyribose and use the base uracil in place of thymine. Once the mRNA strand is synthesized, it undergoes several modifications. In eukaryotic cells, these include the addition of a protective cap on one end, the addition of a tail of adenine bases on the other, and the removal of non-coding regions called introns. The remaining coding segments, known as exons, are spliced together to form the final mature mRNA transcript. This mature mRNA now carries the information required to build a protein and exits the nucleus through pores in the nuclear envelope. Translation is the next stage, and it occurs in the cytoplasm. The mRNA strand attaches to a ribosome, a complex molecular machine composed of ribosomal RNA and proteins. The ribosome reads the sequence of nucleotides on the mRNA in sets of 3, known as codons. Each codon specifies a particular amino acid, the building blocks of proteins. Transfer RNA, or tRNA, molecules ferry amino acids to the ribosome, each tRNA bearing an anticodon that matches a codon on the mRNA. The ribosome facilitates the bonding of each new amino acid to the growing polypeptide chain, constructing the protein one segment at a time, in a process that continues until a stop codon is reached. The order of amino acids in a protein determines how it will fold into a specific three-dimensional shape, and this shape dictates the protein's function. Some proteins serve as enzymes, catalyzing chemical reactions with precision and speed. Others act as structural components, forming the cytoskeleton that supports the cell's shape or contributing to the strength of tissues, such as skin, muscle, and bone. Still others function as signaling molecules, transporters, antibodies, or components of the immune system. Despite their diversity, all proteins begin with the same fundamental process: decoding genetic information. Because of the central role of proteins, the expression of genes is tightly controlled. Not all genes are active in all cells, and not all genes are active all the time. Some are only turned on during specific stages of development, while others respond to environmental cues such as temperature, stress, or nutrient availability. Regulatory proteins, enhancers, silencers, and epigenetic modifications all play roles in fine-tuning gene expression. In this way, cells can maintain their identity and respond dynamically to the ever-changing internal and external conditions they face. Interestingly, the same gene can sometimes give rise to multiple protein products. This is possible through a process called alternative splicing, in which different combinations of exons are joined together to form distinct mRNA transcripts from the same gene. This greatly increases the diversity of the proteome, the full set of proteins that a cell or organism can produce, without requiring an equivalent expansion of the genome. In humans, it is estimated that more than 90% of genes undergo some form of alternative splicing, enabling complex functionality from a limited genetic repertoire. Mutations in a gene sequence can alter the protein that is produced. A single base change may result in a different amino acid being inserted into the protein, potentially affecting its function. Sometimes this change is benign. Other times it can render the protein non-functional or even harmful. These alterations can lead to a range of genetic disorders. In some cases, such as with sickle cell anemia, a single base substitution changes one amino acid in a critical protein, altering its behavior and leading to significant physiological consequences. Proteins also interact with one another and with other molecules in intricate networks. Signaling cascades, metabolic pathways, and gene regulatory networks are all composed of proteins communicating through complex biochemical interactions. These networks are often robust, capable of compensating for disruptions, but they can also be vulnerable to failure if key components are compromised. Understanding these networks is a major focus of systems biology, which aims to map the entire web of molecular interactions in a cell or organism. The relationship between genes and proteins is sometimes described as the central dogma of molecular biology. DNA makes RNA, and RNA makes protein. But modern biology recognizes that this is not a one-way street. Feedback loops exist in which proteins can influence gene expression, RNA molecules can regulate other RNAs, and environmental factors can reshape how genes are used. The once linear model has evolved into a dynamic, circular system of regulation and response. Scientific efforts to decode and manipulate this system have led to the rise of synthetic biology, a field that aims to design new biological systems or reprogram existing ones. By assembling synthetic genes and controlling their expression, researchers can engineer cells to produce medicines, degrade pollutants, or perform novel tasks. This is possible because, at its core, the relationship between genes and proteins is programmable. The code is universal, and its logic can be harnessed, redirected, or even rewritten. Understanding this relationship also allows for the development of personalized medicine. By analyzing a person's genome, doctors can identify mutations that affect how drugs are metabolized, how diseases progress, or how an individual's immune system responds. Allows for more precise treatments and targeted therapies that minimize side effects and maximize effectiveness. Proteins produced by specific genes can also serve as biomarkers, helping to diagnose conditions or monitor treatment responses. In agriculture, gene-protein knowledge has been used to develop genetically modified crops that are resistant to pests, tolerate drought, or produce higher yields. These applications reflect how deeply intertwined gene function is with biological performance, and how altering that function can have wide-reaching effects. While these interventions are sometimes controversial, they highlight the fundamental truth that genes, through proteins determine much of what life does. Even in forensics and ancestry research, the gene-protein connection is foundational. DNA sequences encode the traits that distinguish one individual from another. The specific versions of genes we carry, called alleles, influence the proteins our bodies make and thus the observable characteristics we express. These genetic markers can be used to match DNA at crime scenes, trace family lineages, or reconstruct human migrations across history. The process by which a gene becomes a protein is not just a technical phenomenon. It is the very essence of biological life. It is how a single fertilized cell becomes a complex organism. It is how the same set of instructions can produce the diversity of life on Earth. It is how thoughts emerge from neural networks, how wounds are healed, and how memories are encoded in the brain. Without proteins, genes would be silent. Without genes, proteins would have no guide. Together, they form a partnership that sustains every living cell. And still, within all this precision, there is space for variation, adaptation, and evolution. The translation of gene to protein is faithful, but not rigid. Life thrives not in perfect replication, but in controlled imperfection. Small changes ripple through the biological system, occasionally giving rise to new forms, new capabilities, new paths. This quiet machinery, cellular, chemical, relentless, has no awareness of its elegance. Yet it is within this system that life grows, adapts, and endures. To realize that you are the result of such a system, that your skin, your voice, your memories are shaped by proteins that were shaped by genes, invites a kind of reverence. The molecules do not know they are alive, but they create the conditions in which you can wonder, love, and think about your own existence. From code to function, from instruction to experience, the path is both mechanical and miraculous. In every heartbeat, in every breath, the silent reading of your genetic code continues. Chapter 4. Inheritance: How Traits Are Passed Through Time. The continuity of life across generations is one of biology's most profound phenomena. It is the quiet thread that connects parents to children and through them to the future. This continuity is made possible through inheritance, the transmission of genetic material from one generation to the next. At its core, inheritance is the process by which organisms pass on their genes, and with them, the traits and tendencies encoded within those genes. This passing down of biological information ensures the survival of species, the emergence of variation, and the evolution of complexity over time. Inheritance operates through the medium of DNA. Every cell in your body, with the exception of a few specialized types, contains a complete set of your genome. But when it comes to reproduction, only a specific type of cell carries the instructions that will be passed on, the gametes. In animals, these are the sperm and egg cells. Unlike most other cells, gametes are haploid, meaning they contain only one set of chromosomes, half the amount found in a typical body cell. In humans, that's 23 chromosomes in a sperm and 23 in an egg. When these cells unite during fertilization, they create a diploid zygote with a full set of 46 chromosomes, half from each parent. Each chromosome carries hundreds to thousands of genes, and each gene may exist in different forms, called alleles. The particular combination of alleles you inherit from your parents determines many aspects of your physical traits, such as eye color, hair texture, blood type, and even predisposition to certain health conditions. Some traits are influenced by a single gene with distinct dominant and recessive alleles, while others are shaped by the interactions of many genes working in complex, often subtle ways. The principles of inheritance were first systematically studied by Gregor Mendel in the 19th century. By crossbreeding pea plants and observing how traits such as seed color and pod shape appeared in successive generations, Mendel discovered consistent patterns. He observed that traits are determined by discrete units, what we now know as genes, and that these units are inherited in pairs, one from each parent. He also noted that some traits could mask the presence of others. A dominant allele could determine the visible trait, even if a recessive allele was also present. The recessive trait would only appear if both inherited alleles were recessive. These simple yet profound insights became the foundation of what we now call Mendelian genetics. However, not all traits follow Mendel's clear-cut patterns. Many traits exhibit incomplete dominance, where the offspring's phenotype is an intermediate between the two parents. Others show codominance, where both alleles are expressed equally, such as in the case of the AB blood type. Some genes have multiple alleles in a population, and others interact with one another through epistasis, where the expression of one gene depends on the presence of one or more modifier genes. In reality, inheritance is rarely as tidy as Punnett squares might suggest. Beyond these classical patterns, polygenic inheritance plays a significant role in shaping complex traits. Characteristics such as height, skin color, and intelligence are influenced by the additive effects of many genes. Each gene may contribute only a small amount, but together they create a continuum of possibilities across the population. These traits are also influenced by environmental factors, which further blur the distinction between genetic potential and actual outcome. A person may inherit genes for tall stature, but poor nutrition during childhood can prevent that potential from being fully realized. This interplay between genes and environment is a defining feature of human development and diversity. Inheritance also occurs at the level of chromosomes. During the formation of gametes, a process called meiosis, homologous chromosomes pair up and can exchange segments in a process known as recombination or crossing over. This shuffling of genetic material creates new combinations of alleles, increasing genetic variation among offspring. Each gamete receives a random assortment of chromosomes, meaning that siblings, except identical twins, inherit a different mix of genetic material from their parents. This randomness is a central feature of sexual reproduction, contributing to the uniqueness of each individual. Mutations are another mechanism that introduces variation into the inherited genome. These are changes in the DNA sequence that may occur spontaneously during replication or as a result of environmental exposures, such as radiation or chemicals. While many mutations are neutral or even harmful, some confer advantages that may be favored by natural selection. Over generations, these beneficial mutations can spread through populations, driving the gradual evolution of new traits and adaptations. There are also non-Mendelian forms of inheritance that involve genetic material outside the nucleus. Mitochondria, the energy-producing organelles within cells, contain their own small circular genome. Mitochondrial DNA is inherited maternally, passed exclusively from mother to offspring. This form of inheritance has proven useful for tracing maternal lineages and studying evolutionary relationships. Similarly, in some species, the inheritance of certain traits is influenced by epigenetic factors, chemical modifications that affect gene expression without changing the underlying DNA sequence. Some of these modifications can be passed to future generations, suggesting a form of biological memory that extends the classic genetic transmission. The field of population genetics studies how alleles are distributed within populations and how their frequencies change over time. It incorporates factors such as mutation, gene flow, genetic drift, and selection. Genetic drift, for example, refers to random fluctuations in allele frequencies that can have a significant impact in small populations. Gene flow occurs when individuals migrate between populations, introducing new alleles into the gene pool. Natural selection favors traits that enhance survival or reproduction, gradually increasing their prevalence. These processes, acting over thousands or millions of years, shape the genetic landscape of entire species. Our ability to study inheritance has expanded dramatically with advances in DNA sequencing. We can now trace the ancestry of individuals with high precision, identifying the contributions of various populations to a person's genome. Genome-wide association studies allow researchers to identify genetic variants linked to specific traits or diseases. These insights are deepening our understanding of human health and guiding the development of personalized medicine. Modern technologies have also enabled pre-implantation genetic diagnosis, which allows embryos created through in vitro fertilization to be screened for specific genetic conditions before implantation. While these techniques offer potential for reducing the burden of inherited diseases, they also raise ethical questions about the limits of human intervention in inheritance. The possibility of selecting embryos based on genetic traits, not just for health, but for characteristics like intelligence or physical appearance, has sparked debate about the future of reproductive choice and genetic equity. Inheritance is not just a biological process. It is also deeply intertwined with identity. The knowledge of where we come from, biologically, culturally, ancestrally, can shape how we understand ourselves and our place in the world. Genetic inheritance influences our physical appearance, our susceptibilities, and even aspects of our behavior. But it is not deterministic. The same genome can give rise to countless expressions depending on environment, chance, and choice. Every individual is a mosaic of inherited possibilities. The cells in your body carry fragments of DNA that stretch back through the lives of your parents, grandparents, and distant ancestors, through migrations and chance unions and quiet exchanges of chromosomes. That a single fertilized cell, containing half the DNA of one parent and half of the other, can grow into a thinking, feeling, self-aware being is one of nature's quiet miracles. Through inheritance, life not only preserves itself, but diversifies, adapts, and evolves. From the first replicating molecules in ancient oceans to the vast array of species alive today, genetic inheritance has served as both a thread of continuity and a canvas for innovation. It is a process that transcends the individual, linking all living things into a shared biological history. And perhaps, when you look in the mirror, you are not just seeing yourself, but reflections of thousands of lives that came before you. Coded in genes, whispered through time, shaping the color of your eyes, the sound of your voice, the rhythm of your heartbeat. You are the present expression of a story that began long before you and will continue long after, written not in words, but in molecules quietly passed from hand to hand across the generations. Chapter 5. Mutations, when the code changes. The genetic code is remarkably stable. In each cell, DNA replicates with astonishing precision, ensuring that the instructions for life are faithfully transmitted from one cell to the next, and from one generation to the next. But no system is without error. From time to time, a small change slips through. A single letter in the genetic script might be substituted, inserted, deleted, or rearranged. These changes are known as mutations, alterations to the DNA sequence that can have consequences ranging from the invisible to the profound. They are one of the most fundamental sources of biological variation, shaping everything from the shades of flower petals to the course of evolution itself. Mutations are not inherently harmful. They are simply changes. Whether a mutation turns out to be beneficial, neutral, or detrimental depends on its context. What gene is affected, where in the gene the mutation occurs, and how that change influences the function of the resulting protein, if it influences it at all. Some mutations have no discernible effect. These are called silent mutations, often occurring in regions of DNA that do not code for proteins, or in codons where the new sequence still specifies the same amino acid. Others may lead to small changes in a protein structure that are either tolerated or subtly influence the cell's behavior. Mutations can occur spontaneously during DNA replication. Though the cellular machinery is highly accurate, incorporating proofreading and error correction functions, it is not perfect. With trillions of cell divisions occurring in a human lifetime, even rare errors can accumulate over time. Mutations can also be induced by external agents known as mutagens. These include ultraviolet radiation from the sun, chemicals such as those found in tobacco smoke, certain viruses, and even high-energy radiation like X-rays. These agents can damage DNA, introducing breaks, altering bases, or interfering with the replication process itself. The consequences of mutations depend on their type and location. A point mutation changes a single base pair. If this occurs within a gene, it may alter a single amino acid in a protein. Depending on the role of that amino acid in the protein's structure or function, the effect may be negligible or significant. In some cases, such mutations lead to misfolded proteins, reduced activity, or loss of function. A well-known example is sickle cell disease, which is caused by a single base substitution in the gene for hemoglobin, altering one amino acid and causing the protein to form abnormal shapes under low oxygen conditions. Insertions and deletions, where one or more base pairs are added or removed, can disrupt the reading frame of a gene. This type of mutation, known as a frame shift, often results in entirely different amino acid sequences downstream of the mutation site. The outcome is usually a non-functional protein or premature termination. Larger mutations such as duplications, inversions, or translocations affect broader stretches of DNA and can disrupt multiple genes at once. These structural changes can have dramatic effects, especially if they occur in genes critical for development or cell regulation. In the context of reproductive cells, sperm and egg, mutations can be passed on to offspring, becoming part of the inherited genome. These germline mutations are the basis for genetic disorders as well as for evolutionary change. Somatic mutations, on the other hand, occur in non-reproductive cells. They are not inherited, but can affect the health and function of the individual. Cancer is one of the most studied outcomes of somatic mutations. It typically arises from multiple mutations in genes that control cell division, DNA repair, and programmed cell death. As these mutations accumulate, cells may grow uncontrollably, evade normal regulatory signals, and form tumors. Despite the risks they pose, mutations are essential to life. Without them, evolution would not be possible. Natural selection acts on genetic variation, favoring mutations that enhance survival or reproductive success. Over time, these mutations become more common in a population, driving the emergence of new traits, adaptations, and even new species. In this way, mutations serve as both the engine of biological innovation and the source of its fragility. They provide the raw material from which complexity arises, but they also introduce uncertainty into the system. Mutations can also be harnessed intentionally in research and biotechnology. Scientists use mutagenesis, deliberately inducing mutations, to study gene function, model diseases, or develop new traits in crops and microorganisms. Site-directed mutagenesis, a more precise approach, allows researchers to introduce specific changes into a gene to investigate the effects of particular amino acid substitutions or regulatory modifications. These tools have become essential in molecular biology, expanding our understanding of protein structure, enzyme activity, and genetic control. In medicine, identifying mutations has become a cornerstone of diagnostics. Advances in DNA sequencing allow clinicians to detect mutations linked to inherited disorders such as cystic fibrosis, Huntington's disease, or BRCA mutations associated with breast and ovarian cancer. Knowing whether a person carries such a mutation can inform treatment decisions, guide preventive measures, or assist in reproductive planning. Whole genome sequencing can also reveal variants of uncertain significance, mutations whose effects are not yet well understood, but may provide clues about disease susceptibility. Pharmacogenomics, the study of how genetic variation affects drug response, also depends on detecting mutations. Certain mutations can influence how a person metabolizes a medication, whether they respond to a specific therapy or whether they are at risk for adverse reactions. This knowledge allows for more personalized treatment plans, tailoring medicine to the genetic profile of each patient. Not all mutations occur randomly. Some regions of the genome are more prone to mutation than others, due to sequence characteristics, structural features, or exposure to specific mutagens. These hotspots can become focal points for genetic instability. In certain cancers, for example, specific genes are frequently mutated across many patients, suggesting a common pathway of disease progression. Understanding why certain regions are more mutable can help scientists identify vulnerabilities in the genome and design strategies for prevention or intervention. Epigenetic changes, while not technically mutations, can influence mutation rates and gene expression. Methylation of DNA, modification of histones, and chromatin remodeling can affect the accessibility of genes to the transcriptional machinery or to DNA repair systems. In some cases, epigenetic changes can increase the likelihood of mutations by making DNA more susceptible to damage. Conversely, changes in gene expression caused by mutations in regulatory regions can influence an organism's phenotype without altering the protein coding sequences themselves. Transposable elements, segments of DNA that can move within the genome, are another source of mutation. Often described as jumping genes, these elements can insert themselves into new locations, disrupting genes or altering gene regulation. While most transposable elements are now inactive, remnants of their activity are scattered throughout the genome, and some remain capable of movement. Their influence on genome evolution and diversity is still an active area of study. Over long time scales, mutations accumulate in the genomes of populations. By comparing genetic sequences between species or populations, scientists can estimate the rate of mutation and reconstruct evolutionary relationships. These molecular clocks allow researchers to trace the divergence of lineages, understand the history of migrations, and explore how organisms have adapted to different environments. Mutations, once random accidents, become signposts of shared ancestry and transformation. From a structural standpoint, the genome is remarkably resilient. Redundancies in the genetic code, error-checking mechanisms, and the presence of multiple gene copies help buffer against the effects of mutations. Many organisms have evolved sophisticated DNA repair systems capable of correcting damage before it leads to lasting changes. Yet the system is not flawless, and it is this imperfection that gives rise to the richness of biological diversity. Even within an individual, mutations are ongoing. As cells divide, especially in tissues with high turnover, small changes accumulate. Most of these are harmless or corrected, but over a lifetime, the genetic landscape of the body subtly shifts. This mosaicism means that not all cells in your body are genetically identical, even though they originated from the same zygote. The longer we live, the more mutations accrue, and their effects, whether benign, beneficial, or harmful, become part of the quiet narrative of our cellular history. To live, then, is to be in dialogue with change. Every heartbeat, every breath, every exposure to the environment is a chance for the genetic script to shift, however slightly. Most changes will be silent. Some will ripple outward into traits, conditions, or differences. A few, over the expanse of generations, may redefine what life can be. The genome is not static. It is a living document, continuously edited by time, experience, and chance. And perhaps, in some quiet way, this mutability is part of what makes life so beautifully uncertain. We are not exact copies of those who came before us. We are not fixed points in an unchanging design. Instead, we are improvisations, variations on a theme written in molecular ink. Each of us carries a few new lines in the great story of life, unnoticed by most, but there nonetheless. A reminder that even in the most carefully preserved code, there is always room for something new. Chapter 6: Epigenetics, the genes you don't express yet. As we continue this gentle journey into the language of life, we arrive at a subtle but powerful realm, one that doesn't alter the letters of the genetic code, but instead governs how that code is read. If genetics tells us what is possible in the blueprint of life, then epigenetics tells us what is actually built. It is the science of gene regulation, revealing how identical genomes can lead to vastly different outcomes depending on when, where, and how genes are expressed. Epigenetics is the study of the regulatory system that overlays our DNA, modifying activity without changing the sequence itself. Every cell in your body, whether it becomes a skin cell, a liver cell, or a neuron, contains essentially the same DNA, yet each performs vastly different functions. The difference arises not from the genes themselves, but from which genes are turned on or off in each context. This selective gene expression is largely controlled by epigenetic mechanisms, which determine accessibility to the genetic code and influence the levels at which specific genes are transcribed. At the heart of this regulation lies the structure of chromatin, the complex of DNA and proteins that organizes the genome inside the nucleus. DNA wraps around histone proteins to form nucleosomes, which further coil and fold to create higher-order structures. Whether a gene is available for transcription often depends on how tightly its surrounding chromatin is packed. Tightly condensed regions, known as heterochromatin, are generally silent, while more relaxed regions, called euchromatin, tend to be actively transcribed. One major epigenetic mechanism is DNA methylation. This involves the addition of methyl groups, simple chemical tags composed of 1 carbon and 3 hydrogen atoms, to specific DNA bases, typically cytosines followed by guanines, CpG sites. When these methyl groups are added near a gene's promoter region, they can block transcription factors from binding or recruit proteins that repress transcription. In essence, DNA methylation acts like a molecular dimmer switch, lowering the expression of a gene without erasing it entirely. This modification is stable but reversible and plays a key role in development, genomic imprinting, and suppression of repetitive elements. Another powerful epigenetic mechanism involves histone modification. Histone proteins, around which DNA is wrapped, can be chemically altered at various sites. These modifications include acetylation, methylation, phosphorylation, and ubiquitination, each of which affects chromatin structure and gene accessibility in distinct ways. For example, histone acetylation tends to open chromatin and promote gene expression, while certain forms of histone methylation can either activate or repress transcription, depending on context. These changes don't alter the DNA itself, but they dramatically reshape the environment in which DNA operates. These modifications interact in complex patterns sometimes referred to as the histone code, a hypothesis that combinations of chemical marks form a secondary layer of information, directing the cellular machinery toward activation or repression of genes. Much like musical notes arranged to produce different melodies, histone marks in varying arrangements create unique regulatory outcomes. The study of this histone code has become a key part of understanding how cells orchestrate gene expression with precision. Together, DNA methylation and histone modification form part of the epigenetic code, a dynamic system that orchestrates gene expression across the genome. This code is written and read by specialized enzymes and protein complexes, often described as writers, erasers, and readers. Writers add chemical marks, erasers remove them, and readers interpret them to regulate downstream activity. Through this multi-layered system, cells respond to internal signals, environmental stimuli, and developmental cues with extraordinary flexibility. Epigenetic processes are particularly important during early development. As a fertilized egg divides and begins forming specialized tissues, waves of epigenetic reprogramming silence some genes and activate others, guiding cells into distinct fates. Even after development is complete, epigenetic mechanisms continue to regulate processes such as memory formation, immune response, and cellular aging. In fact, certain epigenetic patterns become hallmarks of biological age, leading to the development of so-called epigenetic clocks that estimate aging based on methylation patterns. One of the most fascinating aspects of epigenetics is its potential for inheritance across generations. This idea, transgenerational epigenetic inheritance, suggests that epigenetic marks influenced by the environment can sometimes escape the usual reprogramming that occurs in early embryos and be passed down to offspring. Though still an area of scientific debate, several compelling examples have emerged. In mice, researchers have studied the agouti gene, which affects coat color and susceptibility to obesity, diabetes, and cancer. When pregnant mice were fed diets rich in methyl donors, their offspring were more likely to have healthy brown coats and normal weights, whereas lower methyl availability led to yellow, disease-prone offspring. Remarkably, these differences were not caused by changes to the DNA sequence, but by alterations in DNA methylation patterns at the agouti gene. In humans, a well-known historical example is the Dutch hunger winter of 1944 to 1945, during which pregnant women endured severe famine. Decades later, researchers found that children conceived during this time had different methylation patterns in genes linked to growth and metabolism, compared to siblings conceived before or after the famine. Some of these effects appeared to persist into the next generation, hinting at a memory of environment carried through molecular markings. While these examples remain exceptional rather than the rule, they raise profound questions about biological memory. What if trauma, nutrition, or exposure to toxins could shape not only an individual's health, but the epigenetic state of their descendants? What if resilience itself could be inherited, not in the genes, but in the way the genome is read? To study these complex questions, scientists have developed increasingly sensitive tools for mapping epigenetic changes. Techniques like bisulfite sequencing allow for high-resolution detection of DNA methylation across the genome. Chromatin immunoprecipitation followed by sequencing, SHIP-seq, maps where specific histone modifications occur. ATAC-seq identifies open chromatin regions, those likely to be active. Together, these technologies offer a rich, layered view of how gene regulation is orchestrated in real time and across contexts. Importantly, disruptions in epigenetic regulation are increasingly recognized as contributing factors in disease. Aberrant DNA methylation can silence tumor suppressor genes in cancer or activate oncogenes. Defects in histone modifiers have been linked to developmental syndromes, neurodegenerative disorders, and immune dysfunction. In some cases, the same gene may be expressed differently in two individuals not because of genetic differences, but because of divergent epigenetic landscapes. This insight has opened new avenues for therapy. Epigenetic drugs, agents that target DNA methylation or histone modification enzymes, are being developed and tested in a variety of contexts. Some are already approved for the treatment of certain cancers, such as myelodysplastic syndromes. These compounds aim to reverse abnormal gene silencing or reactivation, restoring a more balanced pattern of expression. Because epigenetic changes are potentially reversible, they offer unique therapeutic opportunities not available with genetic alterations. The future of epigenetics may also include the development of epigenome editing tools, technologies that can target and modify specific epigenetic marks at precise genomic locations. Unlike gene editing, which changes the sequence itself, these tools alter the regulatory state of a gene without rewriting its letters. Such approaches are already being explored to treat genetic disorders, neurological diseases, and even psychiatric conditions, offering a new dimension of control over gene behavior. In parallel, research into environmental epigenetics continues to reveal how external conditions shape gene regulation. Studies on nutrition, Exercise, sleep, pollution, and early life stress are identifying epigenetic signatures associated with health outcomes across the lifespan. In this context, the epigenome serves as both a sensor and a mediator, translating external experience into internal biological responses. As we uncover more about this layer of regulation, it becomes clear that the genome is not a static script, but an interactive, evolving performance. The same set of genes can produce vastly different outcomes depending on timing, context, and regulatory cues. Epigenetics reminds us that what is not expressed is as meaningful as what is. The silenced gene, the unopened region of DNA, the potential that has yet to unfold, these all carry weight in the biology of life. And so, in the quiet realm of epigenetics, we find a profound metaphor that we are not only what we inherit, but also what is nurtured, remembered, and restrained. That expression can be dynamic, selective, and shaped by experience. That within each of us lies a vast reserve of potential, some expressed now, some waiting in silence. The genes you don't express are not lost. They are resting, listening, waiting for a signal you may never consciously send, but which your body, or perhaps your environment, already knows how to give. In that stillness, there is flexibility, and in that flexibility, a soft kind of hope that the future need not be dictated entirely by the past. Chapter 7. The Human Genome, Reading the Book of Us. The human body, with its billions of cells and remarkable complexity, operates under a set of finely tuned instructions encoded in a molecule that is only nanometers wide. These instructions are written in DNA, a molecular language composed of 4 letters A, T, C, and G, repeated in different combinations over more than 3 billion base pairs. When these sequences are taken as a whole, across all chromosomes and all genes, we refer to the collection as the human genome. This genome is not just a database of instructions for building and maintaining a human being. It is also a record of evolutionary history, a source of biological diversity, and a framework that is reshaped by both internal dynamics and environmental influence. Understanding the human genome is a quest to decode not only how we function biologically, but how we came to be and what makes each of us uniquely ourselves. The term genome encompasses the entire set of genetic material in an organism. In humans, this includes the DNA found in the nucleus of almost every cell, as well as a small but significant amount of DNA found in mitochondria. The nuclear genome is organized into 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes. Each of these chromosomes carries a long, linear strand of DNA tightly wrapped around histone proteins, which helps compact the genome while also regulating accessibility for transcription. Mitochondrial DNA, in contrast, is circular and inherited exclusively from the mother. Though it comprises a much smaller number of genes, its role in energy metabolism makes it essential to life. When the Human Genome Project was launched in 1990, scientists set out to map the entirety of the human genome. It was an ambitious undertaking, requiring the coordination of laboratories around the world and years of effort. The primary goals were to determine the complete sequence of base pairs, to identify all the genes contained within that sequence, and to make this information available for further research and medical progress. The first draft was published in 2001, and a more refined version was completed in 2003. Yet even after mapping the sequence, many questions remained. What exactly qualifies as a gene? How many genes are there? And how does the genome operate as a whole system? Initial estimates predicted that humans would have over 100,000 genes, but the final tally revealed a much smaller number, somewhere between 20,000 and 25,000 protein-coding genes. This was a surprise, especially considering that far simpler organisms like nematode worms or fruit flies have similar numbers of genes. This finding highlighted that biological complexity is not merely a function of gene quantity, but also of how genes are regulated, how they interact, and how they give rise to multiple products through processes like alternative splicing. A major revelation from sequencing the genome was how little of it actually codes for proteins. Protein coding sequences make up only about 1 to 2% of the genome. The rest, once dismissed as junk DNA, includes introns, regulatory elements, repetitive sequences, and non-coding RNAs. Although these regions do not code for proteins, many of them play vital roles in controlling gene expression, organizing chromatin structure, and maintaining genomic stability. This non-coding majority is now seen as a dynamic and essential component of genome function. One of the challenges in interpreting the human genome lies in the complexity of gene regulation. A single gene might be regulated by multiple enhancers located thousands of base pairs away, or even on different chromosomes. Regulatory elements can act in concert or opposition, responding to cues from within the cell or from the environment. These interactions form a network of gene control that is not linear, but rather resembles an intricate web, where changes in one part can ripple through the system. Modern tools such as chromatin immunoprecipitation, DNA footprinting, and high-C sequencing have helped researchers map these interactions, but a full understanding of gene regulation remains an ongoing pursuit. In addition to regulation, the genome is shaped by structural variation. While every human shares more than 99.9% of their DNA with any other human, the remaining variation, about one difference in every 1,000 base pairs, accounts for a substantial amount of genetic diversity. Some of these differences are single nucleotide polymorphisms, or SNPs, which can affect gene function or be used as markers for studying disease susceptibility. Other variations include insertions, deletions, inversions, and duplications, all of which can alter genome architecture and influence phenotype. Copy number variations are a particularly important category of structural variation. These involve segments of the genome that are present in different numbers in different individuals. In some cases, these duplications or deletions encompass entire genes, influencing traits such as immune response, drug metabolism, or neurological development. The ability to detect and analyze these differences has improved significantly with advances in high-throughput sequencing and computational analysis, allowing for increasingly personalized insights into the genome. Understanding the human genome also involves studying how genes are expressed over time. Gene expression is not static. It varies by tissue type, developmental stage, circadian rhythm, and external stimuli. Techniques such as RNA sequencing and single-cell transcriptomics have enabled researchers to measure expression patterns with remarkable precision. These studies reveal that even genetically identical cells can exhibit different behaviors based on changes in gene expression, epigenetic modifications, and local cellular environment. The genome, then, is not just a static code, but a responsive system, tuned continuously to the needs of the organism. The study of the genome also extends beyond individual genes to larger patterns and relationships. Comparative genomics, for example, compares the human genome to those of other species, revealing conserved regions that have remained unchanged over evolutionary time. These conserved sequences often indicate essential functions, such as those involved in development or cellular maintenance. By comparing genomes across species, scientists can reconstruct evolutionary trees, identify shared ancestry, and trace the emergence of new traits. The genome also carries imprints of human history. Population genetics has used genome-wide data to trace migrations, admixture events, and population bottlenecks. Ancient DNA studies have recovered genomes from Neanderthals, Denisovans, and early modern humans, revealing interbreeding events and shared alleles that persist in modern populations. Genomic data can also be used to explore selection pressures. identifying genes that have been favored due to disease resistance, environmental adaptation, or dietary changes. One of the most transformative developments in genomics is the ability to sequence individual genomes rapidly and affordably. What once took years and billions of dollars can now be done in a matter of days for a few $100s. This accessibility has ushered in the era of personalized genomics, where individuals can learn about their ancestry, traits, and health risks from a simple saliva sample. While this information can be empowering, it also raises ethical and privacy concerns. How should genetic information be stored, interpreted, and shared? What responsibilities come with knowing one's genetic predispositions? In clinical settings, genome sequencing is being used to diagnose rare genetic disorders, guide cancer treatment, and identify carriers of inherited conditions. In many cases, a single mutation identified through sequencing can explain a previously mysterious illness, enabling targeted therapy or informed reproductive decisions. In oncology, tumor genomes are sequenced to identify mutations that can be matched with specific drugs, a strategy known as precision medicine. The goal is not only to treat disease more effectively, but to prevent it, using the genome as a predictive tool. Yet despite our increasing mastery of genome sequencing, many regions of the genome remain poorly understood. Highly repetitive regions, structural variants, and sequences rich in guanine and cytosine can be challenging to assemble and interpret. Recent advances, including long-read sequencing technologies, have begun to resolve some of these areas, culminating in efforts such as the telomere-to-telomere (T2T) consortium, which aims to produce a truly complete human genome sequence. The genome is also a frontier for new biological concepts. Synthetic biology seeks to write new genetic code, designing genes, circuits, and even entire genomes from scratch. Gene editing tools like CRISPR-Cas systems offer the potential to correct mutations, modify traits, or eliminate disease-causing elements at the DNA level. These technologies promise enormous benefits, but they also compel us to ask fundamental questions about identity, inheritance, and the ethics of biological modification. As we continue to explore the genome, it becomes clear that it is not merely a sequence of instructions. It is a living document, shaped by evolution, tuned by experience, and passed from generation to generation with both fidelity and variation. It is the basis for life's diversity and the foundation for modern biology. And yet it remains incomplete, not in its sequencing, but in our understanding of its full meaning and function. The human genome, when read as a whole, is neither a fixed destiny nor a perfect map. It is a flexible architecture. It provides guidelines, not guarantees. It enables the unfolding of human life, but it does not dictate the details. Between the sequences of nucleotides and the reality of being human lies a space filled with possibility, shaped by development, environment, choice, and chance. And perhaps the most extraordinary realization of all is this: the instructions that formed your heart, your hands, your voice, and your thoughts were quietly waiting inside a fertilized cell, written in silence, yet capable of becoming everything you are. The genome is not just a book of biology. It is the story of how matter learned to reflect on itself, to look outward at the stars, and inward at the strands that made it possible. Chapter 8. Genetic Engineering, Editing Life Softly. For most of human history, we observed the mechanisms of life from a distance. We watched traits pass from parent to child, witnessed the slow evolution of plants and animals, and recorded the patterns of disease without truly understanding their origins. But with the advent of molecular biology and genetic technology, we moved from observers to participants, no longer limited to reading the code of life, but now capable of writing it. Genetic engineering marks one of the most significant turning points in the relationship between humanity and biology. It is the practice of directly altering an organism's DNA, modifying its instructions to correct, enhance, silence, or insert new traits. And though the tools are powerful and the outcomes sometimes dramatic, the core of the process remains remarkably elegant, a quiet reshaping of molecules, base by base, within the code that governs life. At its essence, genetic engineering involves the manipulation of nucleic acids, either DNA or RNA, to change the genetic makeup of an organism. This can be done by adding new genes, deleting or silencing existing ones, modifying regulatory elements, or correcting mutations. These changes can be made in somatic cells, which affect only the individual being treated, or in germline cells, which allow changes to be passed on to future generations. The ability to perform these alterations with precision has evolved steadily over the past several decades, guided by innovations in molecular tools and techniques. Early forms of genetic engineering relied on recombinant DNA technology. This approach, developed in the 1970s, enabled scientists to cut and paste pieces of DNA using enzymes called restriction endonucleases and ligases. Segments of DNA could be isolated from one organism and inserted into another, usually through the use of plasmids, small circular DNA molecules that can replicate independently within bacterial cells. These engineered plasmids became the workhorses of genetic research and the first tools for producing proteins like insulin, which could be manufactured in large quantities using modified bacteria. From these early steps, the field expanded to include more complex forms of genetic manipulation. One approach, known as transgenesis, involves introducing foreign genes into the genome of an organism. This technique has been widely used in agriculture to create genetically modified GM crops that are resistant to pests, tolerant to herbicides, or enriched with nutrients. Transgenic animals have also been developed for research, medicine, and industry, ranging from mice that model human diseases to goats that produce pharmaceutical proteins in their milk. While these developments demonstrated the potential of genetic engineering, they also revealed its limitations. Traditional methods often involved random insertion of DNA, leading to variable expression and potential disruption of other genes. The process lacked precision. What scientists needed was a tool that could target specific locations in the genome with surgical accuracy. This need was met with the emergence of gene editing technologies, methods that could make precise changes to DNA sequences at specific genomic sites. Among these, the CRISPR-Cas9 system has become the most widely known and used. Originally discovered as part of a bacterial immune defense against viruses, CRISPR allows for targeted editing by guiding a protein called Cas9 to a specific sequence of DNA using a complementary RNA guide. Once bound, Cas9 introduces a double-strand break at the target site. The cell's natural repair processes then take over, either joining the broken ends back together, which can introduce small errors, or allowing scientists to insert new genetic material at the break site. The beauty of CRISPR lies in its simplicity, efficiency, and adaptability. Unlike earlier tools which required custom proteins for each new target, CRISPR requires only a change in the guide RNA sequence to redirect its activity. This flexibility has revolutionized genetic research, making it possible to edit genes in virtually any organism, from bacteria to humans, with unprecedented speed and accuracy. CRISPR is not the only tool in the genetic engineer's toolbox. Other systems, such as zinc finger nucleases and talons, transcription activator-like effector nucleases, also allow for targeted genome editing, though they are generally more complex to design and implement. More recently, advances in base editing and prime editing have pushed the boundaries even further. These technologies enable the precise alteration of individual DNA bases without introducing double-strand breaks. Such methods are less disruptive and offer new possibilities for correcting mutations that cause genetic diseases. The applications of genetic engineering span nearly every area of biology and medicine. In agriculture, Genetically modified organisms help increase crop yields, reduce pesticide use, and improve food security. In environmental science, engineered microbes are being developed to break down pollutants, recycle waste, or capture carbon. In medicine, gene therapies are being explored to treat a range of conditions, from inherited disorders like cystic fibrosis and muscular dystrophy to acquired diseases such as cancer and viral infections. One of the most successful examples of gene therapy involves the treatment of severe combined immunodeficiency, CID, a rare genetic disorder that leaves affected individuals without a functional immune system. Using viral vectors, doctors have delivered corrected copies of the defective gene into the patient's own stem cells, restoring immune function in a number of clinical cases. Similarly, gene editing is being investigated for treating blood disorders like sickle cell anemia and beta thalassemia by reactivating fetal hemoglobin production or correcting the underlying mutation in hematopoietic stem cells. The potential of genetic engineering is perhaps most profound when it comes to the prospect of germline editing, making changes to the DNA of embryos, eggs, or sperm that will be inherited by future generations. While this remains a highly controversial area, experiments have already shown that such edits are technically possible. The implications are vast. Not only could genetic diseases be prevented before they manifest, but it might also be possible to enhance certain traits, such as resistance to disease, or even aspects of cognition and physical ability. These possibilities raise deep ethical questions. Who decides what should be edited and why? What are the long-term consequences of altering the human germline, not just for individuals, but for the human species? How do we draw lines between therapy and enhancement, between treating disease and pursuing perfection? And what responsibility do we bear for the unintended consequences of changes made at the most fundamental level of life? Many researchers and bioethicists advocate for a cautious, regulated approach. The scientific community has called for transparency, global dialogue, and international standards before germline editing becomes a clinical reality. At the same time, the urgency to treat devastating genetic diseases drives continued research into safer and more precise tools. As genetic engineering becomes more integrated into medicine, public understanding becomes essential. Misinformation, fear, and unrealistic expectations can skew perceptions and hinder responsible progress. Education and communication must evolve alongside the science, ensuring that society moves forward with both knowledge and care. The role of regulation is also central. Governments and international bodies are developing frameworks to oversee genetic engineering, balancing innovation with safety, and ensuring that benefits are equitably distributed. Issues of accessibility, consent, and long-term monitoring remain critical as therapies move from the lab into clinical practice. Beyond medicine, genetic engineering is altering how we think about biodiversity and conservation. Scientists are exploring the use of gene drives, engineered genes that spread through populations more quickly than natural inheritance would allow, to control disease-carrying insects or invasive species. In some cases, efforts are underway to revive traits from extinct species, a field sometimes referred to as de-extinction. These applications underscore the profound power we now hold to alter ecosystems, to reshape evolution, to influence the trajectory of life on Earth. Despite its potential, genetic engineering is not a universal solution. Biology remains complex, and even precise edits can produce unintended effects. Off-target mutations, mosaicism, immune responses, and unforeseen interactions with other genes or environmental factors are all active areas of concern and research. The more we learn, the more we recognize the need for humility in the face of life's intricacy. Still, the ability to edit DNA, to gently reach into the code and make a change, is among the most remarkable capabilities science has ever developed. It is not a replacement for nature, but an extension of it, a way of participating in the processes that once unfolded without our intervention. Used wisely, it holds the promise to alleviate suffering, protect ecosystems, and deepen our understanding of biology itself. And somewhere in this quiet revolution, we find a reflection of something deeper, that the molecules we once believed were immutable are in fact open to revision. That life, while resilient and ancient, is not beyond our touch. To edit DNA is not merely to change biology, it is to accept a kind of responsibility. Because in writing the code of life, we do not just alter organisms, we shape futures, we sculpt possibility, and we carry the burden of choosing carefully. In the silence of a lab, under a microscope, a strand of DNA is cut and mended. No sound, no flash, just atoms rearranged. And yet in that stillness, something irreversible begins. A new trait, a new hope, a new story. We are the authors now, not of life itself, but of its next chapters. Chapter 9. Everyday Genetics, from Ancestry kits to cancer tests. Genetics, once confined to the realm of research labs and academic journals, has quietly moved into everyday life. No longer a concept reserved for specialists, it now resides in living rooms, doctors' offices, and even online shopping carts. With the proliferation of at-home DNA tests, personalized health assessments, and genome-based diagnostics, genetic information has become part of the modern human experience. Whether seeking answers about ancestry, risk for disease, or the effectiveness of a medication, individuals today are engaging with their DNA in ways that were unimaginable just a few decades ago. The emergence of direct-to-consumer genetic testing was a turning point in public engagement with genomics. Companies offering Ancestry services use small saliva samples to extract DNA and analyze hundreds of thousands of genetic markers, specific positions in the genome that are known to vary between individuals and populations. By comparing these markers to large reference databases of individuals with known geographic and ethnic backgrounds, the tests can estimate the likely origins of a person's ancestry. These estimates are not absolute truth, but statistical approximations based on genetic similarity and population genetics models. The more comprehensive the reference dataset, the more refined the estimate becomes. Ancestry testing has offered many people an accessible window into their personal heritage. It has connected distant relatives, confirmed oral histories, and in some cases, overturned long-held family narratives. Genetic genealogy, the practice of combining DNA testing with traditional genealogical research, has even been used in forensic science to identify unknown individuals or solve cold criminal cases. By uploading genetic data to shared databases, users can match with genetic relatives, trace maternal or paternal lineages, and sometimes discover connections across continents. While Ancestry insights may satisfy personal curiosity, the medical applications of everyday genetics have far deeper implications. The same technology used to analyze DNA for heritage purposes can also reveal variants linked to health conditions. Some direct-to-consumer tests now offer health reports that assess genetic predispositions to certain diseases, traits, and drug responses. These include single gene disorders such as cystic fibrosis or BRCA-tau-related breast and ovarian cancers, as well as complex conditions like type 2 diabetes, Alzheimer's disease, or cardiovascular risk. However, interpreting this information is not always straightforward. Not all genetic variants are deterministic. Most common diseases are polygenic, driven by many genes of small effect interacting with environmental and lifestyle factors. A person may carry a variant associated with increased risk and never develop the condition, or conversely, develop it without carrying any known risk variant. For this reason, genetic test results must be viewed in context, often with the guidance of genetic counselors or healthcare providers trained in genomics. In clinical settings, genetic testing is becoming a cornerstone of personalized medicine. One of its most impactful uses is in oncology, where genomic analysis of tumors can identify mutations that inform treatment decisions. Tumor sequencing can reveal whether a patient is likely to respond to certain therapies, such as PARP inhibitors for BRCA-mutated cancers or immunotherapies for tumors with high mutational burdens. By targeting treatments to the genetic profile of the tumor, clinicians can avoid ineffective interventions and improve outcomes. Beyond cancer, pharmacogenomics, the study of how genetic variation influences drug response, is gaining ground in everyday practice. Some people metabolize medications too quickly or too slowly due to differences in genes encoding liver enzymes, such as those in the cytochrome P450 family. Knowing this can help physicians adjust drug dosages or select alternative therapies, reducing the risk of adverse reactions or treatment failure. As pharmacogenomic data becomes integrated into electronic health records, such insights may soon guide prescribing decisions as routinely as age or weight. Carrier screening is another area where everyday genetics plays a vital role. Prospective parents can undergo testing to determine whether they carry mutations that could be passed on to their children. This is especially relevant for autosomal recessive conditions, where both parents must be carriers for a child to be at risk. Advances in sequencing technology have enabled expanded carrier screening panels that test for dozens or even hundreds of conditions simultaneously, offering more comprehensive reproductive planning options. Genetics also intersects with lifestyle in subtle ways. Some genetic tests suggest predispositions for traits such as lactose intolerance, caffeine metabolism, sleep patterns, or physical endurance. While these insights are typically not medically actionable, they appeal to people seeking greater self-understanding. Still, the scientific support for some of these trait predictions is variable, and companies offering such reports must balance accessibility with responsible communication of uncertainty and nuance. For all its promise, the use of genetic information in everyday life raises critical questions about data privacy, equity, and interpretation. DNA is uniquely identifying, and its misuse can have far-reaching consequences. Concerns about unauthorized access, data breaches, or third-party sharing of genetic information, especially with insurers or law enforcement, have prompted calls for stronger regulatory oversight and data protection standards. In some jurisdictions, laws like the Genetic Information Non-Discrimination Act, GINA, offer protections against the use of genetic data in employment or health insurance decisions. But gaps remain. Another challenge is the equitable distribution of genetic tools and knowledge. Most genomic databases have historically been dominated by individuals of European descent, which can skew the accuracy and applicability of findings for people from other backgrounds. Efforts are now underway to diversify genomic research, but disparities in access to testing, counseling, and follow-up care persist. Ensuring that the benefits of everyday genetics are shared broadly, rather than reinforcing existing inequalities, will require thoughtful policy, education, and investment. Despite these complexities, genetics has become an integral part of modern life. Newborns are routinely screened for metabolic and genetic disorders. Pregnant individuals may undergo non-invasive prenatal testing, which analyzes fetal DNA circulating in maternal blood. Individuals with a family history of cancer or heart disease are increasingly referred for genetic counseling. Even in the workplace, wellness programs and employee benefits packages sometimes include optional genetic screenings. The quiet integration of genetic tools into health and wellness reflects their growing utility and our growing comfort with their use. The future of everyday genetics will likely involve even deeper integration with digital technology. Mobile apps and cloud platforms are already enabling individuals to manage, interpret, and share their genetic data. Artificial intelligence is being applied to genomic analysis, pattern recognition, and predictive modeling. Long-read sequencing, Multi-omic integration and real-time genomic surveillance are advancing the resolution and scope of what can be learned from a single sample. As data accumulates, personalized risk scores may become as routine as blood pressure measurements or cholesterol levels. Yet with all these advances, caution remains essential. Genetic knowledge is powerful, but incomplete. It can inform, but it does not define. A person's DNA may hint at susceptibility, but it cannot predict how they will live, what choices they will make, or how their environment will shape their future. There is a risk in placing too much emphasis on the genome as a blueprint for life, neglecting the complexity of biology, behavior, and circumstance. And so, everyday genetics walks a delicate line between empowerment and overreach, between insight and determinism. Used wisely, it can improve lives, guide healthcare, and deepen our connection to our past and future. Used carelessly, it can mislead, stigmatize, or distract from more pressing determinants of well-being. Still, there is something quietly poetic about the idea that your saliva, mailed in a simple tube, can reveal echoes of your ancestry, traces of ancient migrations, or risks written into your cells. That a test you take on a whim might uncover a hidden link to someone on the other side of the world, or a reason to schedule a doctor's visit just in time. In this way, everyday genetics is not just about code or chemistry. It is about people. It is about stories that were never fully told and stories that are still unfolding. And maybe, somewhere between the strands of DNA and the choices we make, we find the humbling truth that who we are is never just inherited, but also continuously shaped by how we live, what we know, and how gently we choose to act on it. Chapter 10. The Future of Genetics, Where We Might Go From Here. The story of genetics began with our desire to understand how traits are passed from one generation to the next. What started as the study of simple inheritance has evolved into a sweeping exploration of biology, technology, and what it means to be human. Today, genetics is no longer confined to the abstract world of academic discovery. It has become a daily presence in medicine, agriculture, forensics, and personal identity. But the most transformative chapter may still lie ahead. The future of genetics is not only about improving health or decoding ancestry. It is about reimagining what is possible when we can read, write, and even design the code of life itself. One of the most significant frontiers in genetics lies in whole genome sequencing. As the cost of sequencing continues to fall, the prospect of sequencing every individual's genome as part of routine healthcare becomes more feasible. Unlike current genotyping methods that examine only a subset of known variants, whole genome sequencing captures the complete DNA sequence, including non-coding regions, rare variants, and structural rearrangements. This comprehensive view will allow for more accurate disease risk predictions, the discovery of new disease-associated genes, and a better understanding of how genetic variation contributes to human diversity. With this information, genetic risk profiling will evolve from general population averages to highly personalized assessments. Instead of being told that a condition runs in a family or affects a demographic, individuals will receive risk scores tailored to their own genetic makeup, lifestyle, and environment. These polygenic risk scores, based on the cumulative effect of thousands of small genetic variants, are already being developed for conditions like coronary artery disease, type 2 diabetes, and certain cancers. As their predictive accuracy improves, they may become integrated into routine medical screening, prompting early interventions, lifestyle adjustments, or preventative treatments. Gene therapy is also advancing rapidly. Recent breakthroughs have shown that it is possible not only to treat genetic diseases, but to cure them. By delivering correct copies of genes to patients with faulty ones, or by editing the genes already present in their cells, researchers are addressing conditions that were once considered untreatable. inherited retinal disorders, spinal muscular atrophy, and severe immunodeficiencies have already seen successful interventions using gene therapy. The long-term durability of these treatments is still under study, but early results suggest them may offer lasting relief after a single administration. The next generation of gene editing technologies will go even further. Researchers are now developing techniques that bypass the need to cut both strands of DNA entirely. Base editing and prime editing are two emerging approaches that allow for subtle but precise changes to the genome, capable of correcting point mutations, inserting new sequences, or repairing existing ones with minimal disruption to the surrounding DNA. These systems show promise for treating a broad range of genetic conditions while reducing the risks traditionally associated with double-strand breaks, such as unwanted mutations or off-target effects. In parallel, a field known as epigenome editing is gaining attention. Rather than altering the DNA sequence itself, this approach modifies how genes are expressed by targeting the molecular tags that control gene activity. Epigenome editors use engineered proteins to add or remove chemical groups, such as methyl or acetyl groups, at specific genomic sites. This allows scientists to turn genes on or off without making permanent changes to the underlying code. Such methods could be particularly useful for conditions where dysregulation rather than mutation is the primary issue, offering a reversible and fine-tuned method of genetic control. Synthetic biology, a discipline at the intersection of genetics, engineering, and computer science, is poised to redefine what it means to create life. Rather than simply editing existing genomes, researchers are beginning to design and construct entirely new ones. In 2010, the first synthetic bacterial genome was assembled and inserted into a cell, demonstrating that it is possible to create life forms with entirely human-written DNA. Efforts are now underway to build synthetic chromosomes for yeast and other model organisms. The ability to write genomes from scratch could lead to custom-designed cells for producing vaccines, biofuels, or therapeutic compounds, and eventually, even entirely novel organisms built for specific purposes. In agriculture, the future of genetics is equally transformative. Advances in genome editing are enabling the development of crops that are not only more resilient and productive, but also tailored to specific environments and nutritional needs. Unlike traditional genetically modified organisms, which often involve the insertion of foreign DNA, gene-edited crops can involve small, targeted changes that mimic those that might occur naturally over generations. This distinction may make them more acceptable to the public and more straightforward to regulate. In livestock, similar techniques are being explored to reduce disease susceptibility, improve animal welfare, and increase efficiency in food production. Genetics is also becoming increasingly integrated with other layers of biological information, what scientists refer to as multi-omics. Alongside genomics, fields such as transcriptomics, proteomics, metabolomics, and microbiomics are providing data on gene expression, protein function, metabolic activity, and the microbial communities that live in and on us. The integration of these datasets will allow for a systems-level understanding of health and disease, revealing how genetic variation interacts with molecular networks and external influences to shape outcomes. Artificial intelligence and machine learning will play a central role in making sense of the vast data generated by these technologies. Complex algorithms can detect patterns in genomic data that are invisible to human researchers, identify previously unrecognized associations, and even generate hypotheses about gene function or drug targets. In time, predictive models based on genetics and other data streams could guide personalized treatment plans, simulate disease progression, and help allocate medical resources more efficiently. We may also see the rise of embedded genomic monitoring, devices or implants capable of detecting biological changes in real time. These could include sensors that monitor gene expression in specific tissues, or bio-integrated circuits that respond to early signs of inflammation, infection, or mutation. Combined with wearable technologies and personalized data streams, such tools could revolutionize how we approach preventive care, moving from reactive medicine to a model based on continuous observation and early intervention. As genetics becomes more powerful and pervasive, the ethical, legal, and social implications will become even more pressing. Debates about gene editing, especially in human embryos, remain unresolved. The potential to prevent devastating genetic conditions is accompanied by the risk of misuse for non-medical enhancement, for eugenic agendas, or for inequitable distribution of benefits. The question of who has access to genetic technologies, who controls genetic data, and how consent is obtained and maintained will require constant vigilance and inclusive dialogue. Data security and privacy will also be critical concerns. Genetic data is not only deeply personal, it is also predictive, familial, and persistent. A person's DNA contains information about their relatives, their ancestry, and potentially their future health. Once sequenced and stored, that information cannot be changed. This permanence makes the responsible handling of genomic data a matter of public trust. Policies governing data use, sharing, and protection will need to keep pace with technological innovation. Education will be a cornerstone of the future of genetics. As genetic testing becomes more common in clinical settings, healthcare professionals will need to be trained not just in how to order tests, but in how to interpret them, explain their implications, and guide patients through decisions that may carry significant emotional or medical consequences. Likewise, the general public will need accessible, accurate information to navigate the growing presence of genetics in everyday life. Empowering people with knowledge, while avoiding overhype and fatalism, will be essential for realizing the full promise of this field. In the decades ahead, genetics may also help answer some of the most fundamental questions in biology. How does complexity arise from simple rules? What makes one species distinct from another? Why do some individuals thrive in adversity while others struggle despite advantage? As we peer deeper into the genome and its regulation, we may discover new principles of development, resilience, and adaptation. We may come to understand not just disease, but the architecture of health itself. And perhaps, beyond the technical marvels, the future of genetics will serve as a mirror. As we decode what it means to be human in molecular terms, we are also asked to reflect on what we value, what we want to preserve, and how we want to shape the generations that follow. The ability to read and write the genome is not just a scientific achievement, it is a cultural moment. It tells us that we are not only products of evolution, but stewards of its next steps. In the end, the future of genetics is not only written in code, it is written in the choices we make about who we are, what we share, and how we use the knowledge we now possess. The genome may be a blueprint, but its future lies in our hands. Quietly, patiently, the code waits, and with it, the possibility of healing, understanding, and perhaps even reshaping what it means to live. And now, dear friend, you've wandered through the quiet language of life. Whether you're asleep or simply drifting, know this. Within you is a story billions of years in the making, and tonight you gave it your attention. Rest well, and may tomorrow feel just a little more connected to the miracle that you are.